How's it going, guys? I'm Carlos, and I decided to do my lab video project on how to extract your own DNA. So over the course that 2-week period or week period, I don't know, about learning about DNA, the Punnett squares, and like genetic code, I just find it really interesting about how it pretty much makes us us. DNA is the fundamental and distinctive breakdown of who we actually are as people and what makes us us. So upon learning that information, DNA is a covalently bound molecule in every cell of our body. What its job, what its purpose in the cell is that it carries instructions in order for an organism to develop, survive, and reproduce ultimately. So some fun facts about our DNA is that it is actually 2 meters long, or about 6 feet long, when fully stretched out. When compressed into our cells in our body, it is actually about a nanometer long, which makes it able to fit inside of it and do its job. Now with all that out of the way, let's get to the supplies in which you'll need to extract your own DNA. You'll need rubbing alcohol, dish soap, a stick to extract your DNA to pull it out from the solute. You can use any cup, but I'm using a glass cup so you could actually see the DNA come up to the top. A cup filled with 2 cups of water, any type of table salt, and then a cup to spit your salt to water in. Alright, so what you'll need to do first is you'll need to grab your table salt, and I'm using a spoon for my measuring. So what you'll need to do is you'll need to add about a tablespoon of salt into the 2 cups of water. Our DNA has a sugar phosphate backbone, which is charged. So by adding the salt to the mixture, it basically neutralizes that DNA charge and it makes the molecule less soluble in the water. So once you're done adding the salt to the water and stirring it around, you're gonna drink it and swish it in your mouth for about a minute long. And once you're done with that, you're gonna spit it into your separate cup and let it sit for a little. Once you're done with that, you're gonna add 2 drops of dish soap into your salty water mix, like so. Now it's about 2 drops. So basically what the soap does is that it acts as a separator. So this dish soap, it separates polar and non-polar substances to break from each other. So in this case, we have DNA, which is a polar substance, and the cell membrane, which is a non-polar, which it kind of extracts the DNA and takes it out so that we can see it. After you added the 2 drops of soap, you want to stir it gently because you don't want to agitate it and make a bunch of bubbles. So after you stirred your soap gently in the water, you're going to add about a 1/4 cup of rubbing alcohol to that mixture. About a 1/4 cup. And you're going to stir that and wait about 3 minutes so that it can activate. When you're done mixing it, you could just pour it into a glass cup so that you can see it rise to the top. Just like so. So the rubbing alcohol that's used, it's basically used to precipitate the DNA to make it that white clump form that we can see with the naked eye. So that is basically what it looks like. You can see all the tiny DNA strands. I actually pulled one out right here. It's kind of hard to focus. Well, you see it. So that is basically how you extract DNA from your own self. Thank you.